So hello everyone, let us get going today. So I hope my screen is visible. Okay, let us begin. So let me welcome you all to week 9 Introduction to Developmental Biology PMRF-NPTEL lecture. So if you have been following the previous videos, you do know that the PMRF-NPTEL lectures are designed in such a way so that a PMRF fellow such as myself can interactively solve various course problems on the while interacting with the various students who are opting for the NPTEL course. This not only helps the students to better understand what the course content is of that particular week, but also helps them tackle and know how to approach the assignment problems that are given at the end of the course. So let me introduce myself. My name is Anuvab Chatterjee. I'm a PMRF fellow and PhD scholar under the supervision of Professor Jonaki Sen at the Brain Developmental Biology Lab, Department of Biological Sciences and Bioengineering, Indian Institute of Technology, Kanpur. I am conducting these PMRF NPTEL lectures for the course Introduction to Developmental Biology, and this is the week 9 lecture. In this particular lecture, in this particular week, we have majorly gone through plant biotechnology and plant development and learned how exactly fertilization occurs in plants in order to better understand how in various multicellular organisms the process of development occurs differently. As we know that in animals and plants, the lineage is different, so multicellularity originated differently between both of them. So comparing the similarities and dissimilarities, we can actually better understand the developmental process in multicellular organisms. So let us begin. So our first question of the day is, with respect to formation of salivary glands whose position is controlled by the expression of sex chromosomes reduced, this is a gene that plays a major role in the formation of salivary glands, which of the following statements are false? So for context, SCR gene is expressed in a particular pattern throughout the embryo, and with its expression as well as the competing genes which inhibit its ability to form a salivary gland, the position exactly where the salivary gland primordia will be formed is very restricted. So essentially we have a exact XYZ or Cartesian coordinate at which the salivary gland will be formed, and this is due to an interplay of SCR gene expression as well as genes which inhibit HCR function. So the question asks us which of the following is false regarding this entire formation process. The options are: expression of SCR throughout the embryo leads to the formation of salivary glands in the strip along the dorsal ventral axis. Expression of SCR throughout the embryo leads to the formation of salivary glands in a strip along the length of the embryo, that is, this is the anterior posterior axis. The position of the salivary gland along the anterior posterior axis is determined by SCR expression. The position of the salivary gland along the dorsal ventral axis is determined by DPP, which is the Drosophila analog for BMP, and dorsal expression patterns. And dorsal is another protein that we have looked into majorly while discussing dorsal ventral polarity of Drosophila embryos, and we have seen that dorsal expression ventralizes the embryo. So these are dorsal ventral patterning factors, and the position of salivary gland along the dorsal ventral axis is determined by DPP, and dorsal expression patterns is the final option. So we have to choose which of these following are false. And since this is a round type of question, only one is correct. That is, one is only false among these statements. So anyone will try and answer this. Which one amongst these are false? Okay, so let us go through the solution slide and try again. So this diagram shows us how exactly the two-dimensional coordinate system positions the organ primordia, which means that when various genes are expressed in very minute patterns, as you can see right here, this is an example of how a gene which is visualized using the green color goes along the anterior posterior axis. The one in yellow and blue go along the dorsoventral axis. And as you can see, various points, there are intersections where all of these three are expressed together. So we have points such as these or these where the gene expression is very specific. So the cells in this area will express the purple gene, blue gene, yellow gene, and the red gene. And thus this signifies a very spatially restricted expression pattern of cells, and these cells can go on to form various organs or organ primordia. Similar, this is explained via the SCR activation. SCR, what SCR does is it tries to form salivary gland throughout wherever it is expressed. It is expressed in a dorsal ventral pattern, so shown right here, so up to down. But expression of DPP inhibits SCR, and in the dorsal also inhibits SCR. As we know, DPP is present on the dorsal side, and dorsal is instead present on the ventral side. So because of the intersection of these genes, only this particular area where there is no DPP, no dorsal, and only SCR can form the salivary gland. This is what we mean by the Cartesian coordinate system of gene pattern expression. That is, looking at exactly where various genes are being expressed, the cells which receive signals are very spatially restricted, and thus they only can form. So no cells here, or anywhere else in the diagram can form the salivary gland, just in this position where SCR is present and no DPP and dorsal is present. So using this combination of genes, we can achieve restricted formation of organ primordia such as the salivary gland. So what did we get from here? We got that DPP and dorsal are expressed in a dorsal ventral pattern. SCR is also expressed in a dorsal ventral pattern, and all of these help in the formation of salivary gland. So if we go back to our question, we can start cutting out which of these are true. So SCR is throughout the embryo along the dorsal ventral axis. This is true. We have just seen that. SCR throughout the embryo is along the length of the embryo. That is not true. The length of the embryo is anterior posterior axis, and as we have seen, SCR is only expressed in the dorsal ventral region. So this is obviously the option that we will mark as false. So position of the salivary gland along the anterior posterior axis is determined by SCR expression, as we have just seen, and position of dorsal along the dorsal ventral axis is determined by the DPP and dorsal expression, just as we have seen. So the correct option is number 2. Okay, so anyone wants to ask anything from this? Okay. So we move on and go to the next question. Now we are going into the plant development directly, and this is something that is very basic about plant development. What are the 3 basic tissue systems in plants? So plants have 3 types of tissues, and we have to choose which one. So the options are xylem, epidermis, endodermis, dermal, ground and vascular, xylem, phloem and ground, and cortex, vascular and pericycle. The options are, again, one choice is only correct, as these are round type of options. So anyone wants to try and answer this? What are the 3 basic tissue systems in plants? Thrupati says it is C, xylem, phloem and ground. Arul Mosi also says it is C, xylem, phloem and ground. Anyone else wants to try and answer this? Okay, so let us look at the solution slide itself. So the types of permanent tissues in plants are actually dermal, ground and vascular. So xylem and phloem fall under the vascular tissue. So, but the 3 distinct ones are dermal, ground and vascular. And as the name suggests, dermal is the outermost covering which as our outermost corporal ring, also known as epidermis, same. In case of plants, this can form waxy cuticle in the stem, stomata in leaves, and as well as root hairs on the root. These all are formed from dermal tissues. Next are ground tissues, which help in metabolism, storage, and functions. These include parenchyma, collenchyma, and sclerenchyma. This is something that is we have learned in very basic biology. And finally, there are vascular tissues, which are the conducting tissues, which include the xylem and phloem. So the actual correct answer would be dermal, ground, and vascular. So here is a diagram representing that. The dermal tissue is present on the epidermis or the outer covering of the tree or the plant, and the stomata cover the leaves, like the other pores on the leaves, which help in various gases exchange. Then there are the ground tissues, which help in the metabolism. These are the metabolically active cells present in the plant: parenchyma, collenchyma, and sclerenchyma. And finally, we have the vascular tissue, which help in movement of fluid across the plant. And seeing in cross section, you can look at it in this way: dermal tissue is the outermost, ground is in the middle, and the very center is the xylem and phloem, which are the vascular tissues. So the correct option is dermal, ground, and vascular. In this case, xylem, phloem, and ground. So xylem and phloem combine to give us the vascular part and ground, but we are missing the dermal. So the correct option will be B. Next, we move on and go right into plant development itself. The question is, the ploidy of a sporophyte cell is 2n. What is the ploidy of the spores produced by the sporophyte? Options are 2n, 4n, 1n, and 8n. Only one option is correct. So for context, sporophyte cells, so plants can exist in two distinct forms that are very different from animals. They can have a diploid organism, which is the normal organism that we see, like which can be very easily compared to animals. The diploid organism, which has a 2n number of chromosomes. These 2n produces spores, which are haploid. So these are 1n, obviously. But instead of in case of animals, where these 1n or cells are directly gametes, they may not be gametes. They might be a separate organism, yeah, or a separate phase in the cell cycle, in the life cycle, where the organism can exist in 1n form. So the 1n spores form multicellular structures and can live like that for long. And then they develop into gametes, which are also 1n. And these organisms, this part of the life cycle, which can this stage of the life cycle in which the organism can actually just autonomously develop as a 1n haploid organism, is known as the sporophytic stage. It's known as the gametophytic stage. And the organism, the 2n diploid organism which form the spore, is known as the sporophytic stage. So in the sporophytic stage, the sporophytic cells produce the spores, which are haploid, and these spores will eventually give rise to gametes. And thus this haploid stage is known as the gametophytic stage. So gamete forming and spore forming. 2n is the one that forms spores, it is known as sporophyte. 1n forms gametes, that is why it is known as gametophyte. So the question here is, if the ploidy of the sporophyte cell is 2n, what is the ploidy of the spores produced by the sporophyte? The options are 2n, 4n, 1n, and 8n. Only one is correct. Anyone will try and answer this? Okay, so for hint, since the spores are haploid, so there will be 1n, right? So let us go into the life cycle of an angiosperm, which is a flower producing plant. So this, as I said, is divided into two major parts: the diploid sporophyte generation and the haploid gametophyte. What happens is that the diploid plant formed the various spores or the haploid cells, which will give rise to the gametes. These include the pollens as well as the megasporangium, which contains the embryo sac and the zygote, embryo sac and the oocyte specifically, and the microsporangium, which contains the pollens or the sperm cells. And this formation of the haploid cells is what changes from diploid organism to haploid organism. Now the haploid gametophyte can be present in the form of a haploid organism and can live in this autonomous stage for long. And that is the major difference between animals and plants. So here we can see that the diploid sporophyte forms spores which are haploid or 1n, and these undergo fertilization to reform another diploid organism. Thus, diploid organisms which produce spores or the haploid generation are known as sporophytes. Haploid organism 1n which give rise to gametes which in turn fuse to form diploid embryo are known as gametophytes. And thus the diploid organisms or the sporophytes produce spores which are 1n or haploid, which is what we see normally in animals as well. So if the ploidy of the sporophyte cell is 2n, what is the ploidy of the spores produced by the sporophyte? It will obviously be 1, because the spores themselves are haploid. Is that okay? Anyone wants to ask anything from that? Okay. So we move on to the next question. With respect to the germ cells, what is or are the primary differences between plants and animals? So there are major differences, but we are now only looking towards the germ cell differences. The options are: meiotic division of plant germ cells do not form gametes, whereas animal germ cells do. Unlike animals, plants do not set apart germ cells during embryogenesis. The third option is: there are no germ cells in plants. And the fourth option is: unlike animal germ cells, plant germ cells do not undergo meiosis. So this is a box type of question, so more than one is correct. So anyone will try and answer this? You can go ahead and just try answering the question. It will be, at most it will be wrong, and then we can have a discussion on why is it, and on the other hand it can be very well be correct. So anyone wants to try and answer this? You want to try guessing? Okay, so let us look at the unique aspects of plant development and try to get an answer out of this slide. The unique aspects of plant development, which are different, very different from that of animals, is: the first is plant cells do not migrate. Plants have sporic meiosis rather than gametic meiosis. So spores are undergo meiosis, not gametes. The life cycle, but both of these are germ cells, so germ cells undergo meiosis. The life cycle of most plants alternate between diploid and haploid multicellular stages that we have just seen. Plant germ cells are not set aside early in development. Plants undergo extended morphogenesis. Plants have tremendous developmental plasticity, and multicellularity evolved independently in plants and animals. So the points which are related to germ cells here are exactly the point of meiosis and the fact that germ cells do not set aside, the plants do not set aside germ cells early in development. So the primary differences are these two. Meiotic division in plant germ cells do not form gametes, whereas animal germ cells do. Instead, they form spores. And unlike animals, plants do not set apart germ cells during embryogenesis, but like animals, we have seen in Drosophila development that the pole cells are set apart very early, whereas in plants that does not happen. So the options are 1 and 2. Next, which of the following is the strongest evidence to say that plants have tremendous developmental plasticity? So developmental plasticity means that they can develop in various forms. So the options are in support of this: alteration of generations in single life cycle, that is from diploid to haploid; maintenance of meristem cells throughout the life. Stem cells are the stem cells of plants. Ability to produce lost parts and ability to produce wide-ranging morphologies influenced by the environment. So only one of the four is correct. So what this means is that developmental plasticity means that the same plant or very similar plants, if taken in various contexts, can develop differently. That is what developmental plasticity means. So which of the four actually is the strongest evidence for it? So one, two, three, four. These are round type of questions. So only one of them is correct. So anyone wants to try and answer this? Trupti says it is number B, maintenance of meristem cells throughout the lifetime. Anyone else wants to try and answer this? Okay, so let us look at an example. This is a banyan tree. So we have seen this around us many a times. But these on the left, I would like you to focus on the image in the left. You would see that these banyan tree is something that we have seen that where a banyan tree grows up or various trees that grow up near solid structures such as a wall. And as we can see it, the banyan tree has evolved to go around the structure and forms roots that are above ground and then reach ground from where it can take nutrients, and it is essentially growing up from the wall itself. Whereas a banyan tree, if left alone completely, will form this huge entire canopy which is circular in nature. Instead, this particular plant only will form a canopy that is away from the wall. So even though these are the same plant, they can develop very differently based on the environmental conditions. And that is what is the major point in developmental plasticity, that they can develop even though it is the same organism, depending on the developing environment, it can develop very differently. So we have seen these examples very often. If you have seen climber plants, they, depending if they get a wall or not, they will climb up very high, and they will change their shape and growth pattern depending on the support they get. And if there is no support, they will crawl along the ground itself also. So depending on the conditions given to them, their development changes, and thus that is what is known as developmental plasticity. So the strongest evidence is ability to produce wide-ranging morphologies or shapes influenced by the environment. And the option B, maintenance of male stem cells throughout the lifetime. So maintenance of stem cells is because in animals also we also maintain stem cells throughout our life as well. So you have heard of, if you have heard of bone marrow stem cells that represent, which forms new blood cells. So that is not the strongest evidence for the development plasticity of plants. Instead, it is the ability to change their morphologies influenced by the environment when they are developing. So we move on. The next question is, how many nuclei are present in a fully mature pollen? So this is a very interesting thing because in plants, seemingly having greater number of nuclei per cell is a thing, and is something that we'll discuss further in the slides, and that also occur in a fully mature pollen. So how many nuclei are present in a fully mature pollen? There are four options: 3, 2, 8, and 1. Only one option is correct. I would like at least four. There are four of you. Just go for four options, and at least we'll have like a 1/4 chance of getting correct. So, anyone wants to try and answer this? This is a one type answer question. So, how many nuclei are present in a fully mature pollen? You can at least take a guess. Okay. So let us look at the solution slide. So fertilization in angiosperms occurs. So the male counterpart to the sperms is the pollens in angiosperms, and pollens essentially have the pollen when it is first formed, the immature pollen has two nuclei. One from the cell itself. So how does the pollen look? It looks like a simple cell with its own nuclei, which is known as the tube cell. Inside that cell, there is another cell present, which is known as the generative cell, and it has a nuclei of its own. When the pollen reaches a pistil which is compatible to it, and the tube cell elongates such like this, as we can see right here, to in order to reach the embryo sac. And while this occurs, there is a mitotic cell division that occurs of the generative cell itself. And since the generative cells, the cell forms two cells, and thus we have three nuclei present in the fully mature pollen, two from the generative cell or the sperm cell, and one from the tube cell. So the fully mature pollen has essentially three nuclei, which given in context with animal cells it looks very drastically different. So here we have the pollen producing organ, which is known as the stamen, and when we cut it in a cross section we can see microspores, which will give rise to the pollens. And after they mature they have three nuclei, as you can see right here, and these then go into the wind in order to find a compatible pistil in order to perform the fertilization. But there is one point that we have to keep in mind, that fertilization is not an absolute prerequisite for embryo formation. So even though fertilization does not occur, plant cells can form a embryo, and this occurs in the process known as apomixis, that is no mixing, which is the development of an embryo from haploid eggs or non-meiotic cells. So these are not germ-lining cells, but they can go ahead and form the embryo without any need for fertilization. That is something that is special in plants, which is known as apomixis, development without any fertilization of an embryo without any fertilization. So the correct option is number 3. How many nuclei are present in the fully mature pollen? There are 3. Is that okay? No. So we move on. This is another question based on plant development. Successive divisions of a pollen mother cell are found in dash, and simultaneous divisions are found in dash. So when the pollen mother cell or the microsporangium divides, it can either divide, one cell divides into two, and then two to four. Otherwise, there can also be simultaneous divisions in which before the cytokinesis has occurred, another division has already started, and these are separate based on like successive divisions occur in what cell types of plants, and simultaneous divisions occur in what type of plants. The options are monocot and dicots. Dicots and monocots, just the position is rearranged. Angiosperms and gymnosperms and angiosperms. So anyone wants to try and answer this? Successive divisions of a pollen mother cell are found in which type of plant, and simultaneous divisions are found in which type of plants. Okay, so let us look at the solution slide, and if you can see, move this here while I lose my internet connection. Very nice. Hello, am I audible? Hello, hello, can you hear my voice? Hello. Okay. So, yeah, so as we are talking about, I guess Like we went through the question, right? That went through. So let me go ahead and repeat the question. So successive divisions of a pollen mother cell are found in which type of plants, and simultaneous divisions are found in which type of plants? The options are monocots and dicots, dicots and monocots, angiosperms and gymnosperms, and gymnosperms and angiosperms. And we are about to look at the solutions. So this is the pollen formation, or how exactly the microspores give rise to mature pollens. In case of monocots, there are successive normal divisions that normally we will observe. One cell divides into two with complete cytokinesis, then two divide into four with complete cytokinesis in each stage. There can also be the other way around, in which in the while there is cell division occurring, another cell division occurs without complete cytokinesis, and we get a crossed spindle formation, in which like a middle intermediate stage in which there are three nuclei present with spindle forming in each of them, and this is known as simultaneous type of division, and this occurs in dicots. So in dicots, the cells even before cytokinesis has been completed start dividing again to form the final four cells. And in case of monocots, there is successive type of division that is normally seen. One divides into two and two divides into four with cytokinesis at each stage. So major point is the cytoplasm of these two cells have been separated already, and these four are already separated. Whereas while forming the three cell stage and the four cell stage as well, the all the cytoplasm is already connected, and then cytokinesis occurs. So the cell boundary is divided after the four cells are formed. So the options are successive divisions are formed in monocots and simultaneous is formed in dicots. So we move ahead and look at the next question, which is the term apomixis refers to formation of embryos from the haploid egg without fertilization, formation of embryos from sporophytic cells without fertilization, formation of embryos containing two aneuploidy, formation of embryos lacking endosperm and seed coat. Only one option is correct. Apomixis, that we have just looked at it faintly. Which of the following are correct? So what does apomixis mean? Anyone wants to try and answer this? There is only one type of correct question because there is a round type of answers. Okay. So as we have just seen that fertilization is when the pollen and the pollen cells are find a compatible pistil, form the tube cells and go into the embryo sac where the sperm cells fertilize. Okay, so Trupti has gone ahead and answers. She says the formation of embryos from haploid egg without fertilization is known as apomixis. So yes, so fertilization normally occurs in which the compatible pollen, the sperm cells fertilize the particular macrosporangium cells or the oocyte cells and form the embryo. But as I said, in plants, this is not a prerequisite, and development can go ahead from haploid egg as well. So the haploid egg cell can go ahead and form an embryo without any fertilization occurring, and this is known as apomixis. So apomixis is the formation of embryos from haploid egg without fertilization. You are correct. So anyway, so that was apomixis. Anyone wants to add anything to this? Okay. So the next question is, which of the following will be the consequence if micropyle is not present? So for context, micropyle is this opening right here via which the tube cells enter. Via which the tube cell enters. And there is also the fact that in fertilization plants there is a double fertilization that occurs. So you remember that the generative cell divided into two nuclei. One nuclei will go and fertilize the egg cell, and that will form the new generation, that is the diploid embryo. But the other sperm cell will go and fertilize the central cell's two nuclei. So this is also a point that you have to keep in mind, that in the embryo sac there are 8 nuclei and only 7 cells. The central cell has 2 nuclei, and this central cell will, after fertilization, give rise to majority of the seed. The seed, and this will contain the various nutrient sources for the embryo to develop. So double fertilization occurs, in which one sperm cell goes and fertilizes the egg cell, and the other sperm cell fuses with the central cell, and thus two fertilization occur at once. One with between two nuclei, one with three, and this three nuclei fertilization is thus known as triple fertilization. So that was what double fertilization means. So which of the following will be consequence of if micropyle is not present? Micropyle is the opening through which the tube cell enters. The options are: only one fertilization will occur. Second is both fertilizations will fail. Pollen will not germinate. Pollen tube will not elongate. So only one of them are correct. Anyone wants to try and answer this? If micropyle is not present, which of the following will occur? So will only one fertilization occur? Both will not occur. Pollen will not germinate at all. That is, it will not form a tube, extended tube, or a tube will be formed and it will not elongate. Okay, so this is the blown up picture of the embryo sac. We are describing the various cells that are present in the embryo sac. So we have first, so there are, as I said, 7 cells present. 3 are the antipodal cells. These degenerate as the embryo develop. On the other hand, we have the synergids and the egg cell. The egg cell is the one in the middle, and the synergids are on either side of it and help in fertilization of the egg cell and the sperm nuclei. Next, there is the central cell, which has 2 polar nuclei. So this is one cell that contains 2 nuclei, and as I said, this occurs in plants, and this fuses with the second sperm cell to form the seed and the various nutrient sources for the developing embryo. And this is the micropyle through which the tube cell will enter. There are other various parts, including the integuments, which cover the entire embryo sac. So if the micropyle itself is not formed, then the embryo there is no access for the tube cell to enter the embryo, and no question of any fertilization will occur. Because in order for the nuclei to reach the embryo, it needs to enter via the tube cell and the micropyle. So there is no entry, and thus both fertilization of the egg and the sperm and the polar nuclei will fail. Thus the correct option is both fertilizations will fail. Okay. So let us go ahead. Which of the following is true about synergids? The options are: they act as nerve cells for the egg cell. They prevent fertilization of egg cell by the same plant's pollen, which is something known as pollen incompatibility. They are two of the daughters produced by the meiotic division of the megaspore mother cell, and they are siblings of the egg cell produced through mitotic divisions. So we have a meiotic division forming the cells, or we have the mitotic division. These are the two options. So there is only one out of these 4 that are correct, because these are these are round type of answers. So anyone will try and answer this. Trupti says A. They act as nerve cells for the egg cell. Anyone else wants to try and answer this? Okay. So let us go back and look at it. The synergids actually are siblings of the egg cell produced through mitotic divisions. So if we have, okay, so synergids are formed through mitotic division. So essentially there is one megasporangium cell which undergoes meiotic division first, and it forms four 1n cells. So these are the daughter four cells which are haploid in nature. These undergo mitotic divisions in order to form the final structure that we have seen right here. So the four haploids undergo mitotic divisions to form these 8 nuclei, and thus the egg cell is actually egg cell and the synagogues are related via mitotic divisions, and they do not so synagogues do not act as nurse cells for the egg cell. So nurse cells, if you remember correctly, during Drosophila development we have seen these cells help in development of the embryo directly by pushing various proteins via the cytoplasms and the gap junctions. This does not occur in plants. These cells only help in the one of the functions of them is helping only the egg cell be fertilized via the sperm cell that is entry. They do not act as nurse cell throughout the development as nurse cells actually do during Drosophila development, as that we have seen. Instead, they are siblings of egg cell which are produced through mitotic divisions. Okay, so we go ahead and look at the next question. Choose the correct groups. The group H is group with haploid cells. The group D are group with diploid cells. And we have 5 options. We have to essentially separate them. Which of these are haploids? Which of these are diploids? The 5 cell types are tube cell, generative cell, egg cell, integuments, and sporophytic cells. So we have to just take these 5 and then separate it out into various groups. Like we have to choose which are haploids, which are diploids, and then we have more than one option correct type of answer, in which essentially we have to choose a H and a D, and that can be like only 2 we will choose, either like any combination of the cells and the groups that we put them in. So anyone wants to try and answer this, try to check which of these are haploid and which are diploid. So first I will just ask you, which are the haploid cells that you can see here? Name any haploid cells that you can, like can we identify at least one haploid cell out of these 5? Okay, so let us look at the pollen directly. So we have options are tube cell, generative cell, egg cell, integuments, and sporophytic cell. So these are all related to embryo formation and development. So we'll look at both the pollens and the embryo sac separately to identify which are exactly haploid. So this is the electron micrograph of the pollens, and when we look at it at the pollens themselves, as I said, the pollens will contain 2 nuclei. There is one cell, one big cell, which has its nuclei 1n nuclei, so a haploid nuclei of its own, which is known as a tube cell. Inside this tube cell, there is another smaller cell present, which is known as the generative cell. And this generative cell also contains 1 nuclei, and this is haploid as well. So both the tube cell nuclei and the tube cell and the generative cell both are haploid. Next we look at the embryo sac again. As I said that the entire germ-line lineage of the embryo sac is formed from one megasporangium cell, or the embryo mother cell, or the egg mother cell, and it undergoes meiosis first. So one megasporangium will form 4 1n haploid daughters, and these haploid daughters will undergo mitosis to form 8 nuclei. But since we started with 4 haploids, and we know that mitotic divisions do not affect the number of the n, only meiosis does. Thus from the 4 haploid daughter cells we will get 8 haploid daughter cells. So every one of the cells is shown here, the antipodal cells, the polar nuclei, the egg cells, all of these are essentially haploid. Is that clear? So these everything in the boundary is haploid. So the antipodal cells, the egg cells, the synergids, all are haploids. But the integuments, this is part of the sporophytic cell lineage. That is the mother plant or the plant itself, inside which the embryo is present, that forms the integuments. And thus, all of the integuments are essentially diploid in nature, because these are not germline lineage. These are the comparable to somatic lineage in animals. So the integuments essentially are 2N or diploid, but these all of these are 1N or haploid. So let us first divide. So the tube cell, as we have seen, is haploid, generative cell is haploid, egg cell is haploid. So all of these three, 1 to 3, will lie in group H. So the first option. And integuments, as I said, are diploid because it is part of the parent plant. And sporophytic cells. Sporophytic cells form the spores, and those are diploid. And thus the 4 and 5 lie in the group D, and thus the last option is correct. Sporophytic cells and integuments are diploids, tube cell, generative cell, and egg cell are haploids. Anyone wants to ask anything about this? Okay. Next, we tackle the point known as self-incompatibility. In what type of self-incompatibility pollen fails to germinate even when pollen and egg cell do not share the same allele? So since pollen and the pollens that are available to a egg cell can be varied, there is a very high probability that the same plant or similar plants which are very similar genotypically will go ahead and pollinate the egg cell. It can occur in the same plant itself, that we know that some there are flowers where the male parts and the pollens and the egg cells are formed in the same flower, or the same plant also forms pollens as well as the egg cells. So it is very much possible that the same there is a high chance of self pollination. So but this in turn decreases the genetic diversity that a plant can have. So plants show a specific incompatibility towards self-pollens, or pollens that have various characteristics from the parent plant itself or from its own genotype, and thus these pollens are incompatible to fertilize the eggs. There are two types that are there, and in this particular question, we are asked the self-compatible pollen fails to germinate even when pollens and egg cells do not share the same allele. This occurs in which type? So the options are interspecific incompatibility, intraspecific incompatibility, sporophytic incompatibility, or gametophytic incompatibility. Anyone will try to answer this? Yeah, so we'll go ahead and answer this. So let us discuss the incompatibility itself. As I said, it can be either be gametophytic or sporophytic. In case of gametophytic self-incompatibility, we have is the gamete, like the pollen itself, can have two alleles of the same gene. Say they are so this occurs in the self incompatibility locus. So the locus is known as self incompatibility locus and can have various alleles S1, S2, S3, whatever. Say the pollen, there are from the parent plant had S1, S2 stamen, and thus it formed two pollens, one with the S1 genome, one with S2 in its allele in its genome. When these pollens go ahead to pollinate a specific carpel which has a S2, S3. So the S1 goes to a S2, S3 carpel, so the alleles are different, and thus this tube cell can elongate and fertilize these egg cells. So this is compatible. But instead, if S2 goes to a S2, S3 carpel, where there is matching between the alleles that are present between them, then the pollen will not be compatible at all and it will be rejected. So the S2 pollen cannot fertilize this carbon, and that is known as gametophytic self incompatibility, in which the gametes themselves, the alleles of the present in the gametes themselves are important. So if it is S1 or S2 is important in the pollen itself. But in case of sporophytic self incompatibility, we have that in a similar condition, if sporophytic self incompatibility is present, we will see that both of these cannot pollinate the carpel. And this is very interesting because see, we have S1 here and S2 and S3 in the carpel, but still this is rejected. The main reason is because this S1 pollen also came from a stamen that had S1 and S2, and as we have seen that some part of the pollen is formed from the original sporophytic organism as well. So S1 has an outer covering that contains cells with the S2 allele as well, and that is recognized and that is rejected. So even though the pollen showed compatible genotype with the carpel, since the sporophytic cells covering it had an allele which that is S2, which is again incompatible with the carpel, this S1 is also rejected. So in this case, the rejection is not due to the pollen's genotype, but the parent plant's genotype as well. And since the parent plant is obviously a sporophyte, it is known as sporophytic self-incompatibility. So is that clear? Anyone wants to ask anything from this? Okay. So in what type of self-incompatibility pollens fail to germinate even when pollen and egg cell do not share the same allele? So see, that is the second case in which S1 does not share the same allele with the carpel, but still it is rejected, and it is sporophytic incompatibility. So we move on and look at the next question. Which of the following is or are true about the SUS1 gene? Which of the following is or are true about the SUS1 gene? So the SUS1 gene, so many varied studies were done, and it was seen that they obtained a loss of function for the gene known as SUS1, and they found that in that particular loss of function mutant, the embryo did not progress normally, and they wanted to know why that is the fact. So but that is the experiment that we will be talking about. But which of the following is true about the ACS1 gene is what we want to know in this particular question. The options are: it encodes a dicer-like protein. So if you remember, dicer is so Shriyansh goes ahead and answers it essential for laying down the radial axial body plan for the angiosperm embryo. So we'll look into it in the solution slide itself. But yes, the dicer protein, if you remember, it is important for RNAi. That is the dicer protein after when there are microRNAs. So this is something that we had discussed in the very first lectures. There are microRNAs present in our cells. These microRNA formed very short hairpin structures which are cleaved and then incorporated into the protein known as dicer. If these microRNAs are comparable or complementary to a mRNA that is about to be transcribed, then dicer sits on the mRNA and cleaves it or stops its transcription somehow, a translation. So dicer essentially helps in post-transcriptional regulation of gene expression via the microRNA system. So this is majorly important in case of invading foreign organisms as well as regulation as well. So this essentially could have developed as a defense mechanism. So various viruses we have microRNAs against the genome of various viruses, and when they come in and try to transcribe their own genome, the dicer protein and the microRNAs will recognize it and cleave it and prevent its infection. And thus, dicer helps in protection of the genome as well, and that is very important in plants. So one of the options is SUS1 gene encodes for a dicer-like protein. The next is it is involved in the first asymmetric division of the angiosperm embryo. So the angiosperm embryo, if you remember correctly, angiosperm embryo as it develops, it forms two cells. One is the terminal cell, another is the suspensor cell. Terminal cell will actually give rise to the plant or the developing plant. The suspensor cell helps in orientation of the plant while it develops, and thus it is also very important, and this is the first asymmetric division. So maybe so the option 2 will say that SUS1 is important for that. The third option that Shian chose, it is essential for laying down the radial axial body plan of the angiosperm embryo. So the radial axial body plan, so Trupti says that it is number 1, it encodes a dicer like protein. So we will get to that as well in the solution slide. So this is the third option is the one that Shian chose. It is essential for laying down the radial and axial body plan of the angiosperm embryo. So the radial and axial body plan means what? So as we have many a time discussed that there are three axes in case of animals. These include the dorsal ventral, anterior posterior, and the medial distal axis. So essentially the x, y, and z axes. In the case of plants, we have the two axes which are radial and axial. Radials means from the center towards outward, and axial means up and down. So is sus1 is important for laying down the radial and axial body plan for the angiosperm embryo. And the final one, it suppresses suspensor development in the embryo. So suspensor is the one that we just talked about, that after the first division, two separate structures are formed, terminal cell and the suspensor part. Suspensor helps in the orientation of the embryo towards its food source and helps in its development. Terminal cell forms the embryo itself. So it suppresses the suspensor development in embryo. These are the four options that are ahead, like the four options that we have. So during various experiments, it was seen that they, as I said, went ahead and made a loss of function mutation for the suspensor. So I would like you to look at these particular pictures. So here, this ballooned out part, this is the embryo or the developing embryo that will form the entire plant. So these are probably DIC images. So we can see a bit of 3D effect on it as well. So this will what form the embryo itself, and this long stalk-like structure is the suspense. This helps in the orientation of the cell and everything. But in case of SUS gene mutants, we see that there are like strangely two bulbous structures. So we have the suspensor which will not form any bulbous embryo-like structures, but here instead we have at two ends the bulbous structure will form, which will give rise to the embryo, and thus this was something that was being investigated. So in SUS mutants only this occurs. So in normal cases, SUS gene should be involved in suppressing the development of embryo-like characteristics in the suspensor, and that is why in its loss of functions we see such a phenotype in which the the suspensor also tries to form embryo-like structures. So in normal wild type case, SUS1 should inhibit this particular condition from occurring. So after much study, this particular model was proposed in order to explain the function of SUS gene. What they said is that the embryo itself signals from the embryo suppress the embryonic development in suspensor cells. So there is a particular signal coming from the embryo which prevents the suspensor cells from forming another embryo-like structure, and instead from the suspensor itself signals from the suspensor cells promote embryonic development in these embryonic cells. So we have a feedback loop in which the further the embryo develops, it suppresses the suspensor from forming any embryonic structures, and the suspensor helps in the embryonic structures to be formed. And this, in this case, how exactly ACUS gene helps? The ACUS1 gene or GCL1 gene encodes a dicer ortholog, so a dicer-like protein, and therefore may be involved in generating small RNA signals. So ACUS1 is essentially the dicer-like protein present in these cells. Hello, hello, am I audible again? Hello, yes, okay. So we were discussing this particular part. Did we get to the C part, or so I will go through the this part again then. So by in SS1 mutant, they saw that the embryonic character was developing in the embryo itself as well as the suspensor. And they, after looking at it extensively, they proposed a model in which they said there is a feedback loop occurring, in which the embryo cells signal to suppress embryonic development in the suspensor cells, and the suspensor cells instead signal to promote embryonic development. So further the embryo develop, it keeps on suppressing embryonic development in the suspensor cells, and the suspensor cells instead help in the development of the embryo. And thus this particular thing is via the ACUS1 gene and its gene product. The ACUS1 gene encodes a dicer ortholog, so dicer-like protein, and therefore may be involved in generating small RNA signals. So as we have seen, so a dicer is involved in post-transcriptional regulation of various genes. So it is very well possible that the ACUS1 protein, what it does is in the suspensor cells, it regulates the gene expression of embryonic genes. So even though those genes are there, the ACUS1 protein suppresses their expression, and thus this is the basis of the suppression signal from the embryo, that these genes which will develop the embryonic character are suppressed in the suspense. So the correct option is it encodes a dicer-like protein, ACUS1 gene. It is not involved in the first asymmetric division of the angiosperm embryo, because as we've seen, the effect is seen much later after the suspensor embryo have developed a lot. It is essential for laying down the radial axial body plan of the angiosperm embryo. So this is also not correct, because as we've seen that it does not, is not very much related to the embryo development itself. So the radial and the axial development pattern of the embryo is not affected by it, but instead it affects the suspensor formation, which is separate from the embryo development, and thus that option is also not correct. And suppresses suspensor development in embryo, that is not correct. It suppresses embryo development in suppressor, the entirely opposite one. So it suppresses the signals of the suppresses embryonic development in suppressor cells. So essentially, this is also not correct. So the only option that is correct is it encodes a dicer-like protein. That is the first one. Okay. So these are the references for the particular these particular slides. So there were further references that were given in the slides themselves. If you want to check them out, you can go ahead. And the references are, as always, Introduction to Developmental Biology, the book by Scott Gilbert, the 9th edition. This is the YouTube link that on which all these recorded lectures are uploaded, and you can go ahead and refer back to them. So does anyone have anything to add? If not, then we can end today's session right here. Okay. So thank you everyone for joining. See you next week. Thank you.